Welcome, my dear students. Today we are diving into one of the most essential processes of our life, that is DNA replication. DNA is our genetic material, passes from parents to children and from one cell to another. But before it can do that, it must be accurately copied. In this class, we will explore how DNA uses its original strands as templates, follows the Chargaff's ATGC base pairing rule, and produces new strands of DNA. And we also look at the three models scientists propose to explain DNA replication, that is conservative, semi-conservative and dispersive model of DNA replication. Let's unravel the secrets of life copies itself. Introduction. We know that DNA is the genetic material passed from parents to offspring and from one cell to another cell. For this transfer, the DNA molecule must be copied first, and it needs the process DNA replication. Replication uses the original DNA strands as templates, we know, and DNA follows the Chargaff's rule, that is ATGC rule. Adenine always pairs with thymine and guanine G always pairs with cytosine. So during DNA replication, the two strands separate and each strand acts as parental template. Free nucleotides match with the exposed bases on each template strand using ATGC rules. Hydrogen bonding always ensures correct pairing. Covalent bonds form between the phosphate of one nucleotide and the sugar of the previous nucleotide. Result: the two DNA molecules, each with one old, that is parental, and one new, that is daughter strand. So this is the process. So we know that original template strands guide the synthesis of the new strands. So how the pairing occurs? The purine always base pairs with pyrimidine, and that is AT interactions. There is two hydrogen bond, and there is GG interaction where there are three hydrogen bonds. So this is the image showing the basic process of DNA replication. From a parental DNA, two daughter DNA will be produced, and among each daughter DNA, there is one parental strand and one is newly synthesized strand. So DNA replication model, scientists in 1950s proposed three models to explain DNA replication. First of all was the conservative model. Parental DNA strands stay together and newly made DNA strands stay together. But this model has been obsoleted. Semi-conservative model, each new DNA has one parental strand and one DNA strand. This is the most acceptable and mostly correct model for DNA replication. And the other model, dispersive model, which was also ignored and obsoleted, that is the parental DNA and new DNA are interspersed in both strands. So the key points to remember is here: the DNA replication occurs in a semi-conservative mode, and it was further confirmed by Michelson and Stahl experiment. In our next class, we'll teach you, we'll learn the experimental process performed by Michelson and Stahl. They follow ATGC base pairing rules, DNA, and ensure genetic information is faithfully copied. DNA replication is basically the copy of DNA using parental templates. So it produces two identical DNA molecules. So there are three models were proposed, but the semi-conservative model is the correct and acceptable widely. So to sum up, DNA replication is the process by which cells make exact copies of their genetic material. Each new DNA molecule contains only one old strand and one new strand. This is why the semi-conservative model is the correct one. By following base pairing rules and forming precise bonds, DNA ensures that genetic information is faithfully passed on. Understanding DNA replication is key to unlocking the mysteries of genetics, inheritance, and life itself. Thank you.